Hello and welcome. Today we're diving into a technology that's pushing the boundaries of biological discovery: single-cell sequencing. Think about this. Inside your body right now there are trillions of cells, each playing a unique role. Until recently we could only study them in groups, which blurred out the fine details. But what if we could zoom in and understand each individual cell, almost like reading every page in a massive book instead of just skimming the cover? That's exactly what single-cell sequencing allows us to do. By the end of this video you'll understand what single-cell sequencing is, how it works, the different methods being used today, where this field is heading, and why it matters for everything from cancer research to drug development. Stick with us. This is going to be fascinating. What is single-cell sequencing? Let's start from the top. So, what exactly is single-cell sequencing? In the simplest terms, it's a method of studying the genetic material, either DNA or RNA, of individual cells one by one. Now you might ask, why does this matter? Can't we just study tissues or blood samples as a whole? Well, here's the thing. When you look at a bulk sample, you only get an average of all the cells in that sample. It's like listening to a choir and only hearing the blended sound, but missing out on the unique voices of the soprano, tenor, bass, and alto. With single-cell sequencing, we can pick out those individual voices. We can see which genes are active in each specific cell, how different cell types contribute to health and disease, and how individual cells respond to treatments or environmental changes. For example, in cancer research, this is game-changing. Tumors are not uniform. They're made up of diverse cell populations, some of which might resist treatment. Single-cell sequencing helps identify those resistant cells early. In immunology, it helps scientists understand how immune cells respond to infections or vaccines on a cellular level. And in developmental biology, it uncovers the step-by-step transformations that turn a single fertilized egg into a complex human being. Of course, this level of detail comes with challenges: high cost, complex workflows, and the need for specialized equipment and expertise. However, the depth of insight we gain is invaluable, making this technology a cornerstone of modern biology. Plate-based techniques. Let's dive into one of the original methods: plate-based single-cell sequencing. Imagine a plate, like an egg carton, where each well holds one single cell. Scientists place cells into these wells, and then amplify and sequence their genetic material individually. This method is all about precision. Because the cells are physically separated, we know exactly which cell corresponds to which sequence data. This makes it highly reliable, especially in clinical diagnostics where accuracy is non-negotiable. For example, in cancer diagnostics, if you're looking to identify rare, treatment-resistant cells, plate-based methods give you a clear view. The benefits: high accuracy, robust protocols, and established reliability. However, there's a downside: scalability. Processing thousands of cells with plate-based methods is time-consuming and labor-intensive. It's like making coffee one cup at a time when you really need an entire pot. That's where newer high-throughput technologies come in. But before we get to those, it's worth noting that if you're doing a focused study on a smaller number of cells, plate-based methods are still gold standard. Alternatives here include microfluidic-based methods, which we'll cover next. Microfluidic-based techniques. Now let's scale things up. Microfluidic-based sequencing is a method that uses miniature channels and droplets to capture and process individual cells at high speed. Picture this: thousands of tiny droplets flowing through micro-sized tubes, with each droplet carrying a single cell. Inside that droplet, the cell's genetic material is barcoded and prepared for sequencing. This is like an assembly line for cells: fast, efficient, and capable of processing thousands, even millions of cells in a single run. The advantages are huge. High throughput. You can study large, complex samples like tumors or blood with incredible efficiency. Reduced sample waste. Since droplets are small, you use fewer reagents. Scalability. Perfect for large-scale projects like mapping the human immune system or studying developmental biology in fine detail. An exciting application is in drug development, where researchers need to understand how thousands of individual cells respond to new therapies. Of course, no method is perfect. Microfluidic techniques require specialized training, complex equipment, and careful handling to avoid technical noise. If a lab doesn't have access to microfluidics, An alternative would be combinatorial indexing, which we'll mention later. It achieves similar scalability without relying on droplets. But overall, microfluidics is pushing the boundaries of what's possible in large-scale single-cell research. Non-RNA techniques. So far, we've focused mostly on RNA-based techniques. But single-cell sequencing isn't limited to RNA. There's a growing field of non-RNA single-cell techniques like single-cell ADSeq, which examines how DNA is packaged in the cell. This reveals which parts of the genome are open and active. Single-cell DNA sequencing, which focuses on variations and mutations within the genome itself. Why is this important? Because RNA tells us what genes are being used, but DNA and chromatin accessibility tell us why and how they're being regulated. In cancer research, for instance, mutations in DNA drive the development of tumors. Single-cell DNA sequencing allows scientists to track these mutations as they evolve over time, something bulk sequencing can't do. In epigenetics, ATAC-seq provides insights into how environmental factors might turn genes on or off, impacting health across generations. And let's not forget multi-omics approaches. These combine RNA, DNA, and even protein data from the same cell to build a complete multidimensional view of what's happening inside. The benefits of non-RNA techniques: broader system-level insights, better understanding of gene regulation, and the ability to track genetic evolution. The limitations: higher complexity, and often even higher costs than RNA-focused methods. But the depth of understanding they provide is worth the investment, especially in complex diseases and advanced research. So, single-cell sequencing has truly transformed our ability to study biology at the most fundamental level. Whether you're using precise plate-based methods, high-throughput microfluidic systems, or exploring non-RNA approaches, the insights we're gaining are reshaping our understanding of health and disease. From identifying rare cancer cells hiding in a tumor to mapping the entire landscape of immune responses, this technology is paving the way for personalized medicine and breakthrough discoveries. Of course, challenges remain: costs, complexity, and data analysis hurdles. But as the technology advances, we're seeing improvements in affordability and accessibility, bringing these powerful tools to more labs and researchers around the world. If you're as excited about the future of biology as we are, make sure to hit that like button, subscribe, and share this video with anyone curious about the cutting edge of science. Thanks for watching. Stay curious, and we'll see you in the next one.